Hi everyone, and welcome back to another Blue Rose special episode. So tonight we're going to talk all about forensic pathology through the modern lens. So that would be the DNA era. Uh, Before we jump right into it, it's important to note that these webinars are for educational purposes. They're not meant to be used in practice or in clinic or taken as clinical advice or medical advice, or in tonight's case, especially as legal advice. Um, but that being said, noting that this is all for educational purposes and that I am a fallible human who can very easily say incorrect things, um, although I try my best not to. And if I do, you're welcome to message or comment or email or any of the communicate in any of the multiple ways that you can. Of course, you're welcome to interrupt. I see some of the participants as well. So, with that being said, by all means, if we make mistakes, um, tell us, and we will try our best to correct it. Okay, so forensic pathology. So when I hear forensic pathology, I think of one of those crime shows like CSI or one of those ones. So, but what it is, it's essentially where medicine and biology come together to answer legal questions. So, the job would be to document let's say in the case of a murder, it would be to document the body, interpret injury, even disease, and integrate that with the scientific context to arrive at a cause of death, and where appropriate, manner of death. So, DNA has become the gold standard for identification and linking biological matter to individuals. But it's not a truth machine. It's extremely powerful, it's extremely sensitive. But with that being said, if misunderstood or misused, it can be very dangerous. So, tonight is all about getting it's a sort of model that you can use in an autopsy room, in multidisciplinary meetings, in the forensic science lab, or even in court. Repeating that tonight is all about education, and it's a great entry point, but it is not meant to act as medical or legal advice. Right. So taking a step back, right, into the death investigation pipeline to figure out where exactly pathology sits. So you have the scene and the body, which leads to the autopsy, a lab, interpretation, and then eventually in the court. So there's multiple different disciplines from legal to police to scientific to medical involved in all of this. If you think of a death investigation as the pipeline, um, scene findings guide what you look for at the autopsy, and the findings guide what you sample from there. Lab findings guide what you can conclude, and of course, the final step is to translate all of this into something clear and defensible in a case study that you could present as an example to court. Now, forensic pathology is sort of like the hub where we connect biology to the legal narrative, but we are actually very limited in what's present and what's preserved and what's interpretable. And we'll talk a little bit about how sociology and ancient cultures and all of this can fit into the picture. But it's not exactly the topic for the night, but we can mention where we will mention some archaeological forensics and that type of thing that also comes into play, especially with using DNA, but it's not quite pathology. So, what counts as evidence from a body? Now, the body is a crime scene in itself, but the body is also fragile. So DNA labs can work with and pinhead work with pinhead blood spots, for instance, or saliva on a stamp of a letter. So there's multiple different places where you can get DNA, but only if it doesn't get contaminated or destroyed first. So and even pathology can affect this. So for instance, where you cut, how you handle biological samples, what you swab, how you store it, all of these can make or break the interpretation of the results later on. So, because it's all about contamination control in this sort of chain of custody. So, before before DNA, some forensic methods were less sensitive and sometimes less reproducible as well. And so, but in the era of DNA, the biggest enemy is not actually the perpetrator, so to say. It's the contamination and poor documentation of these things. So it's not just, and it's not just lab contamination, by the way. So uh, actually, the biggest place where it is transfer of DNA materials happening through things like paramedics or staff equipment, or even inappropriately sterile gloves. So it's almost like a scientific audit trail. There are many different places in this whole chain, or whole cascade going from the scene of the crime to the lab to the whole process. A lot of places where you can get contamination. So it's reliable in theory, right? But it's unreliable because of the realistic aspect of it, the complexity of all these things that can happen in the world. So a little bit of a history. So before 1986, forensic scientists had far fewer tools to link everyone or evidence to individuals. Um, some of the tools were actually quite impressive, and but other ones were questionable. Um, but DNA forced the field to really grow, and become really more based on quantifiable, repeatable validation and statistics and the scientific method. So, but it also created a brand new problem, which is that people began treating DNA evidence as the infallible truth, right? So, and this is actually quite interesting. So the DNA turning point where they realized the consequences of treating it as infallible truth is was a case. It was called the pitchfork case. So it's probably one of the most important stories in history. And so it, in a nutshell, it didn't just show that DNA evidence can help convict who is guilty, but it's also very valuable in excluding who is not the right a suspect. Sorry. So yeah. Let me repeat that. So DNA evidence is not only about finding who is guilty, but actually also about finding who is not guilty. And this dual role is the moral center of DNA forensics. So it can protect the innocent, if we interpret it correctly. In plain language, what DNA profiling looks like is not. In previous webinars, we've spoken a lot about whole genome sequencing and exome sequencing, and although there is a place, especially in modern labs, for sequencing, um, the usual forensic setting is a lot more focused on a targeted approach. So that's using highly variable or loci, as some people say, um, and genetic barcodes, and to compare the patterns. So if you think about if the profiles don't match, exclusion becomes quite straightforward actually. So and if they do match, the interpretation becomes statistical and context dependent. So the context is an incredibly important aspect of all of this because, well, we'll get into details of why it's important, but let's just talk about them. The original method of doing these DNA fingerprinting. So the original method, I say original, the older method, it's still used in some places, is called VNTR fingerprinting. So VNTR stands for variable number tandem repeats. Its colloquial name is mini satellites. So it produces, or it looks at relatively big DNA fragments, and then you can use a lab method called the Southern blot to try and analyze these. So so it all started with VNTR, and it's longer repeat regions, specifically in the non-coding areas of DNA that repeat with variable length, um, and not just variable length, but variable length between people or across people. So, and this essentially means that it creates multiple different alleles, so recognizable gene sequences. But VNTR methods um, needed a lot of good quality DNA. So we spoke earlier about the envelope and the stamp and the all of these different places where you can get DNA. Problem with that is that the quality of the DNA and the amount of the DNA is not necessarily sufficient. Um, and especially not in a realistic sense of that's not typical of a real crime scene. So VNTR profiling is a foundational concept, and it is still used, like I said, in some contexts, especially the main context where it's used is paternity testing. Um, but in casework, so forensics, they don't tend to use that, except in there are cases where, for whatever reason, it becomes a more useful method or an additional evidence base. But in general, they don't tend to use that as the standard base anymore. What they do tend to use as the standard base is called STR profiling, right? So STR stands for short tandem repeats or microsatellites, right? It's an extremely similar concept, um, but these STRs are shorter repeats. And so you can use PCR, polymerase chain reaction, to amplify regions with a very, very, very small trace amount of DNA, which is great. So now at a crime scene, if you can find even the slightest little swab of DNA, you're able to amplify it with PCR. So it can be a few cells or old samples or even compromised samples in some cases. So clinically, SDR profiling is why modern forensic labs can actually work with a very minimal biological material to start off with. How the profile works, if you go from, say, from autopsy to lab, so you extract the DNA, of course, then you have a little, very, very small amount of the DNA. Then you amplify it using PCR methods, specifically looking at primers for the SDR loci or loci. Fluorescent primers is, in fact, what they tend to use in a process called capillary electrophoresis. So there's different types of electrophoresis. Ultimately, what electrophoresis is going to come down to is it's sending a electric wave through DNA because DNA has a charge, and so if you send this wave through it, it moves based on size. Um, so, for instance, the one of the most basic versions of this that people do in the lab is called gel electrophoresis, and so there, as the electricity flows through the, the gel, um, the smaller fragments move further than the larger fragments through this matrix. But in the case of capillary electrophoresis, when there's fluorescent probes involved, fluorescent primers involved, there's actually a laser that detects how much they've moved. And then software calls out they're called alleles. So, but basically, that's just how you identify the different variants. So, yeah, so just to repeat that, so the primers are tagged with fluorescent dye after you PCR. Then you have lots of labeled, they're called amplicons. So that's once you've amplified, you've got amplicons. And then a capillary electrophoresis separates the fragment by size. And a detector, so a laser usually reads out the fluorescence as peaks. Um, and this is why sample handling is everything. So contamination or mixtures of other DNAs, um, you can't tell whether it's going to be clean DNA or messy until you get the final graph. It's called an electropherogram, which are these sort of peaks that you can see on the computer. And by then, the damage is done. So then you find out at the end, oh, okay, we've contaminated the sample, or there was no DNA in the sample, or that type of thing. So speaking of the electropherogram, um, let's talk a little bit about how you read that, because that's actually quite important, especially for a clinician who does a little bit of forensic pathology. So when you see an electropherogram, it's tempting to treat it like a fingerprint, but it's not that. It's not that simple. So peaks can be influenced by quantity of DNA, degradation, so the quality of the DNA, mixtures, so other DNA, contamination essentially, and artifacts. Artifacts is when it's usually when there's so you can see this on a computer, when there's uh, if you take the electropherogram, where there's a jumble of different um, peaks in one place, that's usually an evidence that there was an artifact. So that's in the reading. So maybe it could be that the software messed up or the laser messed up, but it's a computational artifact. So the correct mindset with SDRs is that it is strong evidence. You can use it as very strong evidence, but you still have to be interpreted with all the probability and assumptions that come with the context. So we'll take one example there. So the contamination example. So if you're working with mixed samples, um, and why forensic pathology often creates sort of the hardest DNA cases. So um, okay, yeah, we'll take the example. So if you have a mixture, so let's say, well, we can think of a case study. So let's say you have multiple swabs from one crime scene. So you have, say, vaginal swabs, you have fingernail scrapings, and you have objects that are lying around the crime scene. So mixture um, is a case where a lot of people could have interacted with all of these objects, and a lot of people could have interacted with this environment. So as you're taking swabs and more samples, um, it becomes more and more possible that you sampling the biological material of multiple people. So how do you channel for this? How do you focus this on? So there's quite a few ways where you can profile and cut out uh, unnecessary or unwanted groups. One common method that usually goes hand in hand is Y SDR profiling. Um, so which is very useful, but it also does come with a built-in limitation. So Y SDR profiling targets Y chromosomes. So you essentially isolating male, in the case of crime, suspects, or male, yeah, there's male victims or suspects. So it's powerful, especially in you can think in sexual assault context, because it focuses only on male DNA um, in a mixed background. So if you're swabbing multiple DNA uh, samples, then you can specifically select for which are the male DNA samples, and then you can amplify and use PCR on that and try and identify that. But it doesn't, unfortunately, identify a unique man in the way that autosomal SDR would. It actually only identifies paternal lineage. So the correct statement is often consistent with this is consistent with a paternal line, but you can't say necessarily that it's this individual. Um, and on the flip side of that, so that's the paternal line. You actually get you can actually use mitochondrial DNA for the maternal line. So what you would do there, you would take the mitochondrial DNA, which is actually also sometimes used or often used when the samples are very small or old or degraded. So if you think in mass disaster cases or historical remains, hair shafts, this type of thing. So there have been quite a few cases where um, let's say they take mummies and then they use the mitochondrial DNA because it is a little bit better preserved over time. But the point is because it's inherited maternally, it doesn't recombine as much, which is excellent for the lineage identification with relatives, but once again weaker for unique individualization. So that's the maternal way of identifying things, whereas the YSDR would be the paternal way. So that's yeah, that's one way to single out different things. A pretty common way to also single out um, specific groups would be SNP profiling, single nucleotide polymorphisms. So SNP profiling can be a better fit for severely damaged DNA or degraded DNA as well, because the fragments required can be extremely short. A SNP is a single nucleotide, so it's a single letter. That the trade-off is that you will need many different SNPs or SNP loci to get the same sort of discrimination power statistically that fewer microsatellites or SDRs or mini satellites would have been able to provide. So with a microsatellite, you would be able to use, for example, five, but with and be able to statistically say, okay, this is this individual or has this lineage or whatever. But with SNPs, you're going to need more to get the same level of statistical um, power. So yeah, so but the SNPs actually comes with an interesting other thing that you can do, which is DNA phenotyping. Now, DNA phenotyping is very interesting, very promising, but it's actually also very controversial and not actually usually used in courts because it comes with a lot of context dependent and variable factors that can influence it. So, what is DNA phenotyping? So that's a shift when you go from who is this to what might this person look like, which is fundamentally different. So it can generate leads, especially in cold cases. Um, but it's, yeah, like I said, it's very controversial because it can drive biased policing, especially if handled poorly, and because some claims can definitely exceed public yeah, validation. But it is quite interesting. So you can use, if I give an example, you can use SNP profiling to see, to predict just from biological material what eye color someone has, what yeah, what eye color someone has, or what hair color someone has, what someone's ancestral lineage would be. So you can see how you can sort of start creating a picture or a phenotype. But you can also very clearly see how this can become very controversial. So, what does it actually mean if you have a match with say, SNP testing or VNTR or SDR testing? So, um, this is sort of the courtroom heart of the issue. So, if a suspect does match, um, let's say SDR loci, you must ask, well, how likely is it that it's just a random match, right? So there is always a probability that it's just a random match. So that's where the population allele frequencies and the product rule comes into play. So mathematically, what you do is you multiply the genotype probability across the number or the different loci that you use, the different gene regions, and then the result can become astronomically small. So the probability of it being random, you can calculate, and it becomes incredibly small, and especially the more regions you look at. But that's only if the assumptions actually hold and that the profile is complete and clean. So because we spoke about all of these different ways in which DNA contamination can happen, and there have and has been cases of say purposeful DNA tampering and that type of thing. The other important thing, as always, is of course data and databases. So, uh, databases allow laboratories to compare different profiles across cases and individuals. It's very useful for solving these cases, and they have solved quite a few cases and identified victims this way. But it actually also raises real civil liberty concerns. So who is included in these data, how long are the profiles kept, and how is the data governed? There's a movie, and I unfortunately forget what the movie is, but it's based on a real story. His name is, but it's based on a real story where they were looking for a serial killer and couldn't find any of his DNA on the system, but what they could find was DNA of cousins and distant family members. And so with all of this DNA, they essentially used the population's genetic material to build a case strong enough to be able to identify and find this person, um, which is great because you solved the crime and in that case caught a serial killer. But does also beg the question of well, they didn't consent to giving their data, especially their genetic data, to the police. So is it ethical for them to have used and produced that? And how is this data supposed to be governed? And who is allowed to investigate it. So it becomes with all sorts of interesting cases that have in the past been already been transgressed, and already been used to get a task done. Um, so yeah, because many crimes actually are like that. They don't have DNA, um, or at least not DNA that will answer the key questions. And even when DNA is present, sorry, uh, especially in cases or in countries like South Africa, we deal with a lot of, we're always dealing with a backlog. Um, there's lots of human error, not in South Africa specifically necessarily. There is just does just tend to be human error with something that has such a multi-step or multifaceted systemic pipeline. Um, Especially in transfer, like I said, so DNA can move in very unexpected ways. So because modern methods are so sensitive, um, deliberate tampering is not unheard of. It's not uncommon. So you get these cases where there's synthetic or planted DNA. It's a known conceptual threat, which means that we actually do need to also future-proof for these cases, to include, say, authenticity markers to be able to say that yes, we did find this DNA on the scene, and we can prove it statistically or biologically. We can say yes, this DNA was definitely present. But are we sure that it wasn't put there? Or but are we sure that there isn't a different context that can explain it? So it isn't as straightforward as yes, this test may say there's a very low probability that this isn't this person's genetic makeup or this person's DNA, but of how you interpret that in the context actually makes a massive, massive difference. So um, DNA can be correct and still point to the wrong person. Is essentially what that means. So a DNA match can be analytically correct, um, and still be forensically very misleading. So I think transfer cases teach the deepest rule in forensic pathology, which is DNA can tell you whose biological material is present, but it doesn't automatically tell you how it got there or what it would mean in this context, and it doesn't automatically tell you that this person is guilty. So, uh, and yeah, this is sort of where forensic pathology actually protects the system. So we control, or forensic pathologists control sampling, they preserve context, they flag alternative explanations like transfer usually, and they communicate these and the limits of the DNA technology in court. So, and the reporting language is so important. So the strongest testimony is usually one that's not overconfident or hyperbolic. It's very precise. It's often boring actually, but boring gets the job done in statistically showing, yeah, this is the case, or this is the probability that this is found here, and that this matches that, and it's very mathematical at times, but that's what's needed, precision. So if you leave with a mental model, um, it's that DNA is a powerful tool that lives inside a bigger biological context. In this case, it's applied to everything from investigation to forensic pathology, and that which is essentially connecting the whole story from where the body is found, um, to conviction eventually. Looking at DNA samples and also taking into account different safeguards. Uh, Now, so someone asked earlier, what is the difference between forensic pathology and forensic science? So the big difference is, or the semantic difference, I suppose, the difference is that forensic pathology is medical. So that's a medical doctor, and they do autopsies, they do injury interpretations, and they look at manner of death. Whereas forensic science is a bit broader. So forensic pathology is a form of forensic science, and it looks at not just DNA, but it also looks at toxicology. It looks at trace chemical evidences. It looks at digital or cyber forensics. So pathology is actually only just one branch of it. Um, Now, So yeah, and so I see someone else also asked, does DNA match prove guilt? Um, no, not necessarily. So that's exactly what we spoke about with so DNA matching can prove innocence, or in the sense of it can prove that this person is not a likely suspect, or it can prove guilt in the sense that this person is a likely suspect. But that's all it is. It's a likelihood, it's a probability. And so context, opportunity, timeline, mechanism, transfer alternatives, other evidence, all of these things and should be taken into account. And it doesn't necessarily mean it's very strong evidence, but it doesn't in isolation prove anything necessarily. In fact, so if I list some limitations of DNA profiling, so like I said, there's lots of cases where there just isn't DNA on the crime scene, or the DNA is degraded, or it's uninformative. Um, you also get these cases where there's mixtures or partial profiles, mixtures and partial profiles, and so that would, could be for many reasons, but often it's contamination, or transfer, human error. So labeling errors and switches happen all the time. It's a very common thing in a lab. It's actually strikingly common how often labeling errors can cause mix-ups. And then, of course, DNA is a biological material, so it degrades with time. Um, and we did speak a little bit about the tampering. There have been and will continue to be people who tamper with evidence. It is the real world, and there's many different um, different reasons and different factors to take into account. And though the scientific evidence, yeah, though biological sampling and DNA is great evidence, that's all it is. It's one tiny piece of the puzzle, so it doesn't give all of the answers. So someone asked if wow, okay, so they basically asked, why is phenotyping controversial? Um, so okay, when they say phenotyping, I'm assuming they mean um the sort of crime scene where you use the SNPs to predict different traits or different phenotypes like eye color or skin color or ancestry or that. So it's controversial because of its accuracy. So accuracy limits. There's only so much you can tell. There's privacy issues. Um, and then largely there's also the risk of bias profiling. So even if you've taken a case where you're quite sure that this person has green eyes, there are, or even there are other factors that come into account or could come into account, like epigenetics and expression, or whatever. The suspect might be wearing contact lenses. So there's from the DNA to where you get to the phenotype, there are many different levels in between. There's, like I said, epigenetics, there's metabolomics, um, there's the transcription or the expression, the exome sequencing, sorry, so you're so used to speaking about sequencing. Yeah, the profile of what actually gets expressed. So it is a lot of times that a specific gene sequence does relate to a specific phenotype, and in theory, genotype does equal phenotype. But as any geneticist can tell you, then in practice, you get all sorts of different levels of expression and concepts like penetrance that can affect these type of things, and somatic or morphological, anatomical, physiological things that can make all sorts of changes. So a wild example, but this is an example that exists. There's a genetic mutation that causes the expression of testosterone to be inhibited. And so even if someone were to have an XY sex chromosomes, but the Y chromosome testosterone production is inhibited, anatomically, physiologically, that person would develop to look like a woman. And so that's one example of where you can have bias profiling. And so if you were looking specifically only for people that look like males, you would miss that person in a case. So that's also why it's controversial and it's not as accurate, and it has all of these limits. Um, but it is interesting, and there are there are all these shows that do speak about this sort of um, profiling, and maybe in the future we'll get to a point where you can get an accurate structure of someone's DNA and put it into a machine and create a full holographic image of exactly what they look like. Um, but I don't think that'll ever happen because DNA or genetics is always in balance with the environment. So any expression is always going to be partially DNA, and largely actually the environment or the context that they found themselves in. So yeah, so that's all the questions we have. Um, Let me just see on the webinar itself if we have any questions over there. Uh, no. And so, yeah, with that, that's all that I have for you tonight. But you're welcome to, if you don't have any questions right now, you're welcome to email me. You're welcome to send a message in the chat, and you're welcome to go find us on the YouTube or the Spotify, um, and leave comments there as well. And as I said in the beginning, if you at any point heard any mistakes or even just corrections, not necessarily mistakes, you're more than welcome to bring those up as well. But if there are no further questions, with that, I bid you adieu, and I hope you all have a very good night. Cheerio.